Today I'm going to go through the process of translation. Now you can either use this Google Notes here, Google Slides. This will be on Google Classroom, or I will have my typical sketch notes for translation as well. They will both be up on Google Classroom. I'm going to go through the notes here just to make sure we cover everything. So translation, so this is the process of making protein. So basically we're reading the RNA sequence of the messenger RNA, and from that we're changing the RNA language to an amino acid language. Therefore we're translating it from one to another. And of course translation is done in the ribosome. It's all really based off of the genetic code. So you've probably remember in biology that every 3 RNA nucleotides in the messenger RNA is called a codon. The genetic code is a triplet code. And basically, this genetic code refers to what codon encodes for what amino acid. We'll talk about the start codon, the 3 stop codons. Keep in mind, this is not an overlapping code, meaning that basically you have 3 codes, and then the next 3 codes is the next amino acid, the next 3 codes is the next amino acid. There are more than one code for the same amino acid, so it's considered to be degenerative. That's just basically because if you've got 3 letters out of 4 to make a codon to code for an amino acid, and there's 20 amino acids, there's going to be a bunch of other codes out there, so overlap. So that's the reason for that. And keep in mind that doesn't matter what organism you are, whether you're a mouse, fruit fly, or human, the genetic code is universal. It's another piece of evidence that we all evolved from a common ancestor. All right, so the 20 amino acids are formed from these 64 codons. So make sure you memorize all 64 codons. I'm just kidding. You only need to know one. And the one you need to know is this one right here, AUG, because all proteins start with AUG. So you should know that all proteins start with the codon AUG, just like August is the start of school. And this is a version of the codon chart. There are several different versions of it. I'll link a video to this one of these documents here that you'll have, probably the weekly document, so that you can learn how to read the codon chart in case you do not know how to do that. Normally, I would teach you how to do this, have you do it. If you were getting it wrong, I would watch you do it and check it out. But of course, I can't do that at this point in time. But if you're having trouble with it, make sure you're asking me what it is. So basically, they work by, this is the first letter, this is the second letter, down here is the third letter. So if we're looking at AUG, which is the start, which is down here, if you forget, I would remember AUG is the first codon, so just think how you find AUG. So find AUG on the codon chart. You realize, oh, the A, that's from this side. The A is going to be in this box. The U is from up here, so if we go down, follow the U till we get to the A, we're in this section here. And then we look over to the third letter, G, so we bring the G's at the bottom, so that's AUG here. I do want to point out that in many of these cases, like in threonine's case, the first 2 letters are the same. It doesn't matter what the third letter is. This kind of gives us some mutation insurance, because if that third letter is mutated in the DNA code, it's not going to change the protein. It's going to end up being the same amino acid at that point. So that's the genetic code. Let's talk about translation. It's made up of 3 basic steps that you probably saw this in transcription, and they include initiation. So we start at the first start codon, which is AUG. So we look for that AUG. It's going to code for the amino acid methionine. Happens to be the only code for methionine as well. So that's another way you can find it on the codon chart. Once you got the first one, it's going to start to elongate. So tRNA is going to start bringing in amino acids with its anticodon matching the codon. And that keeps going on until we terminate when we find one of the three stop codons. You don't need to memorize the stop codons because if you can read the codon chart, it will tell you how to do that. All right, so let's look at how this all starts. So here's our RNA code here. You'll notice this part right in front of the AUGs. There's our AUG there. Our part right there, that is referred to as a leader sequence. That basically helps point out to the ribosome here, the small unit of it, where to sit down, where to find that first AUG. So that's going to lie down there. And at that point, tRNA that matches the AUG is going to bring in the methionine. So our first amino acid is now attached. At that point, the second part of the ribosomal unit is going to attach, and it creates two sites, the P site and the A site. And this helps with the tRNAs to make sure everything's lined up correctly, that these two amino acids are lined up correctly so that they'll form the bond. So the ribosome is really acting as an enzyme there, making this bond occur between the methionine, in this case glycine. So in the P site, the methionine is going to be there. In the A site, the tRNA that brings in glycine, which matches to GGA, is going to come in. Once that happens, the ribosome is going to move over one codon. What was in the A site is going to move to the P site. And once the methionine moves out of the P site, this tRNA is going to go out and find a new methionine. Notice that the bond occurred between the methionine and glycine. That's called the peptide bond. Now the A site's open. That leads for the next tRNA to bring in what matches UGA, which is amino acid cysteine. That's going to pull into the A site, which will then attach and form a peptide bond with the glycine. And again, the ribosome then moves over, and the cysteine is going to go from the A site to the P site. Notice it's now peptide bond to the glycine. This tRNA leaves, leaving the A site open to bring in the tRNA for lysine. All right, and this is going to continue and continue on until we hit one of these stop codons. So in the stop codon, in this case is UAA. That does not code for a tRNA, but something called a release factor. So this release factor goes into the P site, causing the whole thing to fall apart. So the two ribosomal units fall apart. The RNA unit will go out, fall apart, and then all of these things, basically, the two ribosome units, the RNA, they could form again and then make another protein. And then here, this protein here is going to go out and start to fold correctly and become a usable protein. And that is all referred to as termination. And one more thing I want to mention before I'm done with these notes is that you don't just make one copy from a piece of RNA. Once that ribosomal units moved far enough away from the beginning of the messenger RNA, from that leader sequence, a new ribosome can attach and move on. And all ribosomes move just like all the other enzymes move, 5' to 3'. And as it moves, you can see the polypeptide chain being made here. Chaperone proteins are going to go in and start folding it. So it's going to actually be starting to fold before it's even done being made. And here's actually an electron micrograph picture of this going on. So you can kind of see the messenger RNA in the middle there. All the little lumps there is ribosomes. And what's coming off there is the polypeptide. And that ends our notes on translation. Take a few minutes and answer the Google Forms to show me that you've watched and understand translation.